This video lesson is going to start with a brief review of the cell cycle, focusing on the differentiating chromatids and chromosomes, and move into a discussion of haploid versus diploid cells, and then a brief review of mitosis as it relates to 2N diploid cells. The cell cycle for a moment. So the cell cycle, you have mitosis. And interphase. Let me finish and then ask. And in interphase, you have S phase, G1, and G2, or G1, S phase, and G2. All right. Now, what happens in S phase of interphase? Synthesis of, not protein, DNA, replication. So during S phase, you've got replication of DNA. All right. Now, when a cell comes out of mitosis, the sister chromatids have been split, right? So I'm going to simplify this with, I'll just use a single chromosome just to simplify it. So let's say these two have resulted from mitosis. Now we're going to look just at this one. What's going to happen during G1? Growth and additional organelles, that type of thing. Then what happens in the S phase? We just said replication of DNA. So at this point, this chromosome here, this is still a chromosome, is going to get a copy or a sister chromatid. This is still, no, because they're exactly the same. Now, I'll show you the colors when we do the other. This is still a single chromosome. It just has a sister chromatid copy. This is a chromosome. This is a chromosome. Oh, I see. That's why you said the colors. Okay, I see what you're saying now. All right, so I'm going to add some color. I don't understand what you meant, but now I do. Now that's with her. So let's say this was our original. Here's our original, and so the copy is represented by the black. All right, but that's still a single chromosome. It was replicated in S phase to have a sister chromatid, so that once it moves through G2 and re-enters mitosis, those chromatids can separate in anaphase.
They don't.
Okay, but the important distinction for this moment, we can review mitosis again later, but the important distinction that I want you to understand for right now, because you have to understand this or you get very confused in meiosis, is that this is a chromosome, this is a chromosome. All right? Okay. The other thing that I think can be confusing, did you have a question, Bradley? No? Okay. So what then is 2N diploid versus 1N haploid? What is the human diploid number? Damien? 46. And the haploid number? You had it. 23. How many chromosomes do your body cells, with the exception of your eggs or sperm, how many chromosomes do your body cells contain? 46. So this is body cells. What do we call the sperm and egg cells?
Gametes.
Sex cells or gametes.
So does N, does the variable N represent the haploid number?
Yes.
Then why does it say 1N haploid?
Because 1 times N is still N. It just helps designate. I've always seen it designated that way. It could just say N, but I've always seen it 1N. I think that's how the book represents it. It's just always seems to be. I guess maybe to make the distinction. All right. Now, taking this back to chromosomes. In order for a cell to be diploid, it has to have homologous pairs, which means it has to have chromosomes represented from both mom and dad, maternal and paternal. Okay. And there really isn't or does not have homologous pairs you could put over there. Yes?
So if the diploid is only one thing, it's not a pair.
You're confusing chromosomes and chromatids. All right, let's look at this again.
Well, I understand what that is. Like, I just don't understand the ends in that.
Okay, so let's look. We'll say we're going to have a simple cell, 2n equals 4. Stereotypically use blue for the male chromosomes and pink for the female chromosomes in this case. Now, is this a 2n cell diploid or 1n haploid? Does it include homologous pairs?
Yes.
Yes. Does it include both maternal and paternal
Yes.
copies for each chromosome?
Yes.
Yes. So this is diploid.
Diploid. So does a human have?
Wait. Okay, now, I'm not going to show crossing over. We're getting ready to look at that in a lot of detail. But I want to show you what happens next, just to make sure that we have this whole diploid haploid thing under control. So I'm not going to show crossing over yet. Let's say the first round of meiosis happens here. What separates in the first round of meiosis?
Just their chromosomes.
Homologous pairs. So,
Could you say their chromosomes? Or is that-
no, that would be incorrect.
Oh, because we're talking about meiosis.
Okay, now, is this cell diploid or haploid? Haploid, yes, because it does not contain both the maternal and paternal copies of the homologous pairs.
It says not to color or shade anything, so how would we show, do we need to show the difference of the paternal and maternal?
When you draw a chiasma, you do need to indicate the different chromosomes, yes, in crossing over. Shading means more not adding shading for dimensional detail in a drawing than providing a distinction between two different things. Does that make sense?
So those two cells are at the end of meiosis 1, right? Like,
Look quickly back at mitosis. Let's go back to our 2n equals 4 example. I'm going to stick to my blue and pink designations. All right, so here's our cell. And this cell gets a copy of the chromosome of each from dad and a copy of each from mom. During the S phase, those replicate. And that leads to, and now it's ready for division.
Not to be confusing, but this mitosis, are we still dealing with parent cells, or is it just...
Otherwise, they're not going to understand the answer to the question how you would have both in the somatic body cells. All right, so we have two homologous pairs here. I'll even underline one, or I'll put centromeres on this one in this color so you can follow this homologous pair. And the other one, I'll put centromeres of this color so you can follow that homologous pair without me having to constantly get the sizes a certain way. So you've got two homologous pairs of chromosomes here. Do you see that? In mitosis, you want to make an exact copy, yes? So in metaphase of mitosis, they line up like this. And then they're going to separate like this. Now, technically, I just want you to be able to follow the pairing. So technically, the centromere's been split, but this helps you follow the pairing. Yes, do you see that? And so then, once those cells split, in the interest of time, we'll just go ahead and take them through telophase and cytokinesis like that. Then when they re-enter the S phase, you've got 2 copies, like that. This is the same homologous pair as this. Do you see that? It's just arranged differently. Now, does that answer the question of how, did that answer your question, Kyle, of how somatic body cells, they still are diploid?
I wasn't thinking about replication.
So that helped?
Yeah.
Okay. Any questions about this now? This was mitosis. I was just showing you how that works to help, again, clarify the understanding of what's in the body cells, why they're diploid, and how that process rolls out. But I did record that, so now we'll have a video of mitosis as well. Yes? A simplified one.
If the diploid cells,
So you stay diploid for the entirety of mitosis. So even when, let me go here, before these enter S phase, you still have the paternal chromosome and the maternal chromosome for each homologous pair.
Right.
That is what makes it diploid. The sister chromatids are just simply copies. This video lesson will cover meiosis 1, meiosis 2, and then nondisjunction as it occurs both in anaphase 1 of meiosis and also in anaphase 2. What's the purpose of meiosis? To make gametes, to make sperm and egg. What does sperm and egg go on to do? What is their fate? To make a full set of chromosomes by doing what? Combining with somebody else's gamete. Yes, that makes a full set. So meiosis, if the point is to make sperm and egg, then you want to end up with half the amount of chromosomes. You got 2 goals here. End up with half the amount of chromosomes and do it in as diverse a way as possible so that natural selection does not wipe you out.
How does it differentiate from the start of meiosis and mitosis? Is it just
It's the way they line up in metaphase and the presence or absence of crossing over. I'll show you that now. Yes?
Just to kind of put in perspective, we have 46 chromosomes in each of our gametes.
No, 23 in your gametes, 46 in all your body cells. Because if you had 46, then they would form 92 chromosomes when they combine with someone else. Does that make sense?
Yeah.
Okay. All right, yes.
are we, in meiosis, is it physically creating the child, or is it?
No, it's making the sperm and the egg.
The sperm to be deployed.
It's making sperm, making eggs.
once it's a baby and you go into
The first, meiosis is split into meiosis 1 and meiosis 2. Prophase 1 is the first step in meiosis 1. Important things that happen here include crossing over. That's a really big one. And also the spindle fibers form, but you're going to find as I move through this, I'm really focusing on what happens with the chromosomes. But it's good for you to know what's going on with the spindle fibers. But as far as the spindle fibers in the nuclear envelope, I'm not as concerned about you having that explained. I think that is pretty readily evident. So we're going to focus on the chromosomes here. Yes?
In regards to the question for the quiz that asks about this process, should we remain focused on what's happening with the chromosomes? Or should we also talk about the spindle fibers as well?
You should talk about spindle fibers as well. Yes. What I'm saying is that I think that you're probably good on that without me focusing on that. I think the chromosomes, what's going on with those is what needs attention from us together in a discussion. Okay, so crossing over is occurring. What is exchanged during crossing over? Alleles. Alleles. The genes are the same. The genes are the same on this chromosome from dad and this chromosome from mom, but the alleles are different. So what is exchanged during crossing over is alleles. What is crossing over occurring between? Homologous pairs, maternal and paternal chromosomes. Yes, not sister chromatids. There would be no point. They're copies. All right, so crossing over occurs between homologous pairs. What's the point of crossing over?
genetic variation.
Genetic variation, genetic diversity, yes. Yes. And the chiasma is this, just the point right here where the chromosomes are actually crossing, and then they're going to exchange alleles at that point. And I try to carry that through with some accuracy. I'm sure it's not exactly, I didn't measure it, but I tried to carry it through so that you could see that. So do you guys understand then what happens in prophase 1? And you're diploid at this point. All right? Do you see there's homologous pairs? Both the maternal and paternal chromosome of the pairs are represented. Okay, so that's diploid. Okay. Then, yes.
What cell does it start from? Like, because it can't start from.
All right, in metaphase, the homologous pairs line up along the equator or the metaphase plate. And so that would be like right here. Yes, the equator or the metaphase plate. Now, remember how when we looked at mitosis, they were lined up in a single file line, basically. All right, this isn't the case here because they're lined up in pairs. That's really important for you to remember. Metaphase 1, in meiosis 1, they line up in homologous pairs. This is where random assortment occurs. What random assortment means is that, I showed it here, so even though crossing over occurred in this way, they can flip and be on this side. You'll also hear this referred to as independent assortment, and what that means is in one homologous pair, you may have the maternal chromosome on one side and the paternal on the other, and on the next homologous pair could be flipped. It's completely random. And see, if you've got that happening each time that meiosis occurs, that imparts a ton of genetic diversity into the gametes that are ultimately going to result. So for metaphase 1, you need to remember, it's very important that you understand and remember that homologous pairs are what line up and separate, and that they can assort themselves. It's random assortment as to how those pairs line up with respect to which one is on which side. So now you've got 2 layers of genetic diversity introduced here in the first part of meiosis 1. Crossing over in prophase 1 and random assortment in metaphase 1. Okay? Does that make sense?
That's what differentiates this specific part of the process from mitosis, in that they're not every single chromosome isn't aligning down the middle. The pairs are aligning.
Yes, that's correct. So that's the major difference between this and mitosis is the lining up of the homologous pairs. And so it's at this point through anaphase that you go from a 2N cell to a 1N cell. Yes, Damien?
So they're always going to be in that pattern, like when they're in that state right there. Or is it?
No. Well, they're always going to line up in homologous pairs in metaphase 1. But random assortment tells us that the way that those pairs line up is random. It's different. Yes, and that imparts a lot of diversity. Yes, Bradley?
If we wanted to actually draw a human one of these, we would have to put 23.
Yes, in order to draw this for a human being, you would need 23 pairs of chromosomes.
So 2N would equal
46.
46.
2N would equal 46. 23 pairs.
Okay, so 2N equals 8. That means you have.
In anaphase, the homologous chromosomes are pulled apart. And I didn't show the bending here, but you can see how they're pulling apart. So you've got the separation of homologous chromosomes in anaphase 1. That's what lines up in metaphase. That's what's pulled apart in anaphase 1 of meiosis, is the homologous pairs are separated. And so then you're going to end up in telophase with something like this. Now, meiosis 1, it's not always going to go all the way through telophase 1 and cytokinesis before meiosis 2 starts. So sometimes you'll see where you can actually see all 4 nuclei and it's still kind of joined in the cytoplasm. Sometimes they will completely separate through cytokinesis before re-entering into meiosis 2. So just be aware of that. Remember, the key here is with the nucleus. Those are the changes that you're looking for. I'm going to show it with cytokinesis because I think it makes it a little easier to see what's going on. So after telophase, if you have full cytokinesis, then you're going to have, and sorry, I was doing a demonstration of nondisjunction and forgot to erase the changes I made last class. There. Yes, clear. So if it goes all the way through cytokinesis, this is what you have at the end of meiosis 1. Are these haploid or diploid? Haploid, yes, yay. I think that, by George, she's got it. You guys watched My Fair Lady? You should if you have not. But it's a great scene about the rain in Spain and how it stays mainly on the plain.
So by telophase, they're haploid.
Why?
Because they aren't paired.
Yes. I just don't want you to try to memorize that was the case. But yes, you're correct. Yes. They're no longer in homologous pairs, so they're haploid. All right. Is that clear? It's good? Okay.
But they're still in sister chromatids, correct?
Yes.
Yes, they still have sister chromatic copies, but they're single chromosomes that aren't in homologous pairs, so they're haploid.
See, I think what I went wrong is I thought homologous pairs meant sister chromatid.
Right. Oh, that's confusing for a lot of people.
And that's why the whole thing was confused.
So in meiosis 2, during prophase 2, the meiotic spindle's going to, or the spindle fibers are going to show up, and you're going to have condensation into chromosomes if they fully decondensed. But again, how far through telophase and cytokinesis it goes is not always the same. Here is a very important distinction between metaphase 1 and metaphase 2. In metaphase 2, you have that single file lineup of the chromosomes. It looks similar to mitosis at this point. Do you see here where so this would be the metaphase plate or the equator, and the chromosomes are lined up along the metaphase plate. You can have random assortment here as well. This is not as important for genetic diversity as random assortment in metaphase 1, but it still does impart diversity because you have had crossing over, which means that you don't have exactly the same thing on one side as the other. So you're going to get even more as these are either this way or a mirror image, depending on how they line up. You're going to get more opportunity for genetic diversity here as well. You see that? Okay, good. Now, in anaphase 2, the sister chromatids separate. And then in telophase 2, you're starting to see the formation, if we were to do, like the nuclear membranes would be forming here. And so the sister chromatids have separated. We were already haploid, so we're still haploid. And then after cytokinesis, you have 4 1N haploid cells, which are what type of cell? Gametes. Sperm or egg.
So we started where we left off, but we have 4 instead of. What did you say?
No, because there's no homologous pairs here, because you want to have 1N so that if these are sperm, then they can combine with an egg to create a 2N zygote, and that's
So
what starts the organism.
Versus infinite number of gametes.
All right, I think I'll make a chart too. All right, now let's look at nondisjunction. Okay, so what happens during nondisjunction?
There's like a chromosome goes in,
It fails to separate.
doesn't separate.
Yes, it fails to separate during what? Anaphase. So in anaphase 1, if we had nondisjunction, and I'm going to lose the colors here because it'll take me all day. If not, you'll be sitting there waiting for me to draw all the colors at each time. So I'm just going to show you how nondisjunction works, I'm just going to do this. Do you see? So during anaphase, in the case of nondisjunction, this pair has failed to separate and are both going to one side. Now, that creates a situation in telophase like this. And so then after cytokinesis in meiosis 1, you get a situation like this. Do you see that? So then, what that's going to do as it carries through meiosis 2, so metaphase, you're going to have an additional one lined up over here. You're going to be missing one over here, additional one separating during anaphase, missing one here, additional here and here, missing one here and here, additional here and here, missing one here and here. If you have nondisjunction in meiosis 1, do you generate any normal gametes? No. No. Is that the only place nondisjunction can occur? No.
Can it occur in
Anaphase
the
2.
anaphase of meiosis 2?
Yes, so let's go back
Which would be the sister chromatid.
and see what would happen if that happened instead. So now we'll look at anaphase of meiosis 2, and this is where the nondisjunction occurred instead. So that would look like this, which would create a situation like this. You see? So if nondisjunction occurs in anaphase 2, do you have any normal gametes? Yes, because these are still normal gametes. All right? Okay. Is that clear? Yes. Okay, perfect.
And is there a way we could get